Welcome to this video on reflex actions and reaction time. We'll explore how your nervous system provides automatic protection and how quickly you can consciously respond to your environment. We will cover four main topics: what reflexes are, the reflex arc pathway, reaction time, and how to measure it with the ruler drop test. A reflex action is a rapid, automatic response to a stimulus, requiring no conscious thought. This makes it extremely fast and effective at protecting the body from harm. Reflexes are important because they are fast, bypassing the brain to minimize damage. They are also automatic, providing reliable, consistent responses every time. Chapter 1 focuses on the reflex arc. This is the pathway an impulse travels from a stimulus to a response through the nervous system. The reflex arc is a specific, short, and fast pathway. The components in sequence are receptor, sensory neuron, relay neuron, motor neuron, and effector. Step 1. Receptors detect the stimulus. Receptors are specialized cells in the skin, eyes, ears, nose, and tongue that detect changes in the environment, like heat, light, or sound. Step 2. Sensory neurons carry the impulse from the receptor toward the central nervous system, or CNS. They have long axons and transmit signals very quickly. Step 3. Relay neurons in the CNS connect sensory neurons to motor neurons. In a reflex arc, the impulse typically passes through the spinal cord, bypassing the brain for speed. Step 4. Motor neurons carry the impulse away from the CNS towards the effector. Remember, sensory neurons carry impulses to the CNS, while motor neurons carry them away. Step 5. Effectors produce the response. Effectors are organs or tissues like muscles, which contract, or glands, which secrete chemicals to carry out the response. This summary table shows each component of the reflex arc, its role, and an example. It's a useful way to quickly revise the pathway. A synapse is the tiny gap between two neurons. Since electrical impulses can't jump this gap, they are converted to chemical signals using neurotransmitters, which then trigger a new impulse in the next neuron. In the hot object reflex, heat receptors detect the stimulus. The impulse goes via a sensory neuron to the spinal cord. A relay neuron processes it, and a motor neuron activates the arm muscle for an instant response. You feel the pain only after the reflex action occurs. Chapter 2 shifts our focus to reaction time. While reflexes are automatic, reaction time measures how quickly you can consciously respond to a stimulus. Reaction time is the time taken from detecting a stimulus to producing a voluntary response. Unlike a reflex, it requires conscious thought and involves the brain. The ruler drop test is a common and simple method to measure reaction time. It involves 3 steps: hold and drop, catch it, and record the distance. For GCSE, remember that a shorter distance the ruler falls means a faster reaction time. For example, 5 to 10 centimeters is considered fast, while 25 to 30 centimeters is slow. Chapter 3 covers factors affecting reaction time. Understanding these variables is important for your exams, as they can slow down or speed up your response. Factors that slow reaction time include tiredness, fatigue, alcohol, and distractions. These affect how quickly and efficiently your brain processes signals and sends responses. Alcohol is a CNS depressant that slows nerve impulse transmission at synapses. This significantly increases reaction time and severely impacts coordination and judgment. Tiredness and fatigue reduce the efficiency of neural signaling. This means slower processing of sensory information and impaired judgment, leading to delayed responses. Reaction time can be improved through practice and training. This strengthens neural pathways, aids motor learning, and leads to measurable gains in response speed over time. Chapter 4 is about scientific reliability. To ensure your ruler drop test results are trustworthy, good experimental technique is essential. To improve reliability, always repeat trials to calculate a mean, control variables, identify anomalies, and use a large sample size. It's crucial to distinguish between the 3 variable types: independent, dependent, and control. In the ruler drop test, the distance caught is the dependent variable. The most common human error is anticipation. To reduce this, drop the ruler without warning, check the grip position, and ensure the catcher maintains eye contact with the ruler. Finally, Chapter 5 reviews key exam concepts. These core facts are frequently tested in GCSE exams. Key exam points include: reflexes are automatic. You must be able to name all 5 components of the reflex arc in order, and a shorter ruler drop distance means a faster reaction time. Avoid common exam mistakes, such as stating the brain sends the signal in a reflex. Also, correctly identify effectors as muscles or glands, and always remember to repeat measurements for reliability. Comparing reflex arc and voluntary action is often necessary. Remember that conscious thought is involved in voluntary actions, but not reflexes. Reflexes are faster, bypass the cerebral cortex, and cannot be consciously controlled, unlike voluntary actions. Test your knowledge with these quick recall questions. Can you define a reflex action and list the 5 components of the reflex arc in order? Think about what a shorter ruler drop distance indicates and name 2 factors that slow reaction time. Ensure you are familiar with all the key vocabulary. You must know the definitions for terms like reflex action, stimulus, receptor, all 3 types of neurons, effector, reaction time, and synapse. To summarize, reflexes are automatic and protective, following the 5-step arc. Reaction time is measurable using the ruler drop test, where a shorter distance means faster response. Remember that factors like tiredness and alcohol slow down reaction time. This concludes our video on reflex actions and reaction time. The next topic will explore the structure and function of the brain, including the cerebral cortex, cerebellum, and medulla.